So good afternoon, ladies and gentlemen. Welcome again to our Together We Succeed webinar series, which we have the privilege to organize together with our colleagues at Miltenyi, Illumina, BioLegend, Merck, Immudex, and Partech. During this webinar series, we want to cover and address different aspects of the single-cell and spatial transcriptomics workflows. My name is Haris Juvali from 10x Genomics, and I will be your moderator for today. All our sessions are live, including a live Q&A session. The webinar is being recorded, and recordings will be shared with you. If you would like to ask a question, please select the Q&A option at the bottom of your screen. We are starting today with an introduction by Stephen Haig, a Senior Science and Technology Advisor at 10x Genomics. We have with us Sarah Bursomano, Product Manager from Immudex, who is going to help us unravel specificity of T-cell immunity using decode dextramers. And we are honored to have with us today Raquel Deering, an Associate Director of Immuno-Oncology Research at Regeneron Pharmaceuticals, who will talk to us about high throughput screening for antigen-specific T-cell reactivities from primary human T cells. Thank you all for being here today. Just before we start, we have prepared 4 questions that should not take more than a couple of minutes to go through, and we would love to hear from you. So here is the first question. Are you running single-cell gene expression experiments? We'll end the poll. So most of you are already running single-cell gene expression experiments. So thank you so much for voting. The second question is, what do you see as the biggest challenge when implementing single-cell methods? Is it sample prep? Is it access to sequencing, data analysis, or any other? So it seems that data analysis is one of the biggest challenges, followed by sample prep. So the third question is, which of the below dextramer technologies have you used in your work? And you can choose multiple ones. It seems that most of you have not used dextramer in your work up to now, and some of you have used MHC dextramers. So this is a correct place to be, I guess, to learn about this technology. And the last question for today is, are you interested in using decode dextramer technology like decode compatible? Excellent. So I will share the results with you. Most of you, yes, are interested. So this is great. Thank you so much for taking the time to answer these questions. And now, Stephen, over to you.
Thanks, Haris. So I just have a little introductory session just to go through a few slides just to set the scene, really. And I'm really excited about this webinar. There's a lot of interesting applications that we can take to with 10x that we'll see today. So I think it will be really pretty interesting. So as we saw from the polls, I think most of you are already pretty familiar with the concept of 10x and how we work. But with all of our products and the ones that we'll talk to today, you know, we start with an input of generally single cells or single nuclei. And then we use the appropriate kit. So today we're mainly focusing on the notion of doing the 5' gene expression because that allows us to do more immune-focused profiling. We make a library and then we can sequence those, generally using the standard Illumina platforms. And then as we see over on the right-hand side, we can do the data analysis. So 10x provides the software to do this. So you can look at gene expression and all of the things that I think we'll talk about today. We can do using our Loop browser software. And what we're really looking to do, as you perhaps can just about see, is in terms of gene expression, we're looking to profile the expression of genes from thousands of cells simultaneously. So potentially thousands of genes from thousands of cells simultaneously. And then what the software is able to do with that data is it's able to split those cells out into cluster analysis, and we could see the different constituent cell types in our sample. So this one is a PBMC sample over here on the right. So 10x works pretty hard to innovate new products, and I think the ethos of what we really try to do is we try to follow what people are looking to do in science that may have real challenges, and we try to come up with solutions that enable people to do that. We started doing sort of standard single cell gene expression back in 2016, and we've had a pretty quick evolution from there. And where we sort of stand at the moment really is we're able to do things like cell surface proteins, which we spoke about last week. We can do things like CRISPR profiling, and we can also do things like antigen specificity. And it's this notion of multi-omics. So we move away from just looking at gene expression down to single cell to being able to tag on these extra moieties as well. So things like cell surface proteins, B cell, T cell receptor sequencing, and those kind of things. And where we've gone to this year is we've gone into looking at targeted gene expression, so being able to reduce down the number of genes that we want to look at. And we've also very recently just updated the 5' gene expression kit, which I'll very quickly mention as we go through. So we really try to innovate in a pretty fast way at 10x. So when we're looking at very much focusing on gene expression using the 5' kit, and the reason that we do that is it allows us to look at VDJ and also we can use T cell sequencing in there as well. So again, what we're seeing here is PBMC cells, so we can split those cell types into the different constituent parts. So we've got macrophages, monocytes, B cells, etc. So we can look at all of that wealth of information in terms of gene expression. What we can also do with this kit is we can look at clonotypes. So for example, here we're able to look at the T cells and the B cells, and we're able to identify the types or the clonotypes that are present, and also really to sort of iterate and count through the number of times that they're represented. And that's a useful process because, if you take a simple example of something like a viral infection, we're actually able to identify the cells that are responding to particular viral epitopes. And that's obviously very a hot topic at the moment with the COVID situation. So as I say, we've been doing the 5' gene expression and being able to do VGJ analysis for a number of years now. And then literally a couple of months ago, we've just released an improved kit. So it works in the same workflow derived way. But what we see in terms of the sensitivity, so over here on the left, we're looking at the sort of median genes that we can see with 1,000 and 10,000 PBMC cells. And over here on the right, looking at UMIs. And what we can basically see, as I'm sure you can see yourself, is that the number of genes that we're able to see is pretty superior using the version 2. So several chemistry tweaks along the way, and that's really allowed us to drive this forward and be able to detect more transcripts per cell, which is obviously what we're interested in when we're doing single cell. And then if we move away just slightly from the notion of looking at gene expression, what we're able to do now, and this is kind of some of the additional moieties that we're able to look at. So we again spoke last week about cell surface proteins along with whole transcriptome, and we can look at things like antigen specificity, which is what we're interested in doing today, and this notion of pair cell receptor sequencing also. So to do the T cell specificity, so to identify T cell populations which are able to bind to particular peptide fragments, as we'll hear today, we work to do this with Immudex. And these, excuse me. So what we can do here is we can barcode effectively, so an oligo barcode onto a dextramer molecule, and we can load peptide fragments into MHC complex, and then we can look very specifically at which T cell populations are able to bind to that, and we can sequence through the barcode, and therefore we can determine ultimately which particular peptide fragment is of interest from those cell types. And we can also do that in conjunction with the feature barcoding technology to also look at cell surface proteins as well. So it's it's a really in-depth analysis that we can do on the immune system. And this is just a simple schematic to sort of show where we've gone internally. We're starting to see customers do this. So we can start with cell suspension. We can, if we want to, flow sort and pull out particular cell types, in this case the T cells. So what we're able to do now is, if we look at all of the different moieties that we can look at, so we can look at single cell gene expression, we can look at cell surface protein analysis, we can identify and isolate, if we want to, those very specific T cell populations that are able to bind to specific peptide fragments, and we can do the VDJ analysis. And the beauty of this is we can do this in parallel on thousands of cells at once, so 10,000 of cells per input, and we can obviously run more than one lane if we want to do that, and we can get single cell resolution on all of these different parameters. So we can really generate an enormous amount of data from our cell populations. And that's really what we're starting to see people do in the world of immunology, this real sort of in-depth study of immunology to really identify what's the driving forces in the response of the immune system, particularly to pathogens and viral insults, for example. So I will say thank you. Hopefully I haven't overrun there, and it's a pleasure to hand over to Sarah from Immudex, who's just going to introduce you a little bit further to Immudex. So thank you very much.
So good afternoon, everyone, and thank you, Harris and Stephen, for the nice introduction. My name is Sara Bursomano, and I'm the Decode Dextramer product manager at Immudex. So over the next 10 minutes, I'm going to talk about the Decode Dextramer technology and its power to unravel the specificity of T-cell immunity. Okay, so let's start with a quick overview about the specificity of the immune response following exposure of the body to a certain antigen. When the body enters in contact with an antigen, like a virus, for example, the organism starts a series of mechanisms that lead to the immune response, involving a variety of different immune cells. So, although all immune responses are similar, each time the body is invaded by different antigen, the exact response is specific to that antigen. And this is the result of the interaction of specific cells activated by the specific antigen. So let's see what happens at the cellular level. When an antigen enters the body, it's taken up by antigen-presenting cells of the immune system to be processed and presented to other immune cells. Small fragments of the antigens are displayed by MHC molecules on the cell membrane and are presented to other cell types of the immune system, like T cells. The T cell receptors, or TCRs, so TCRs that are expressed on the T cell membrane recognize a specific peptide presented in a specific MHC molecule. And this interaction defines the antigen specificities of the T cells. When this interaction takes place, the T cell is activated and proliferate and differentiate, creating a real army of T cells. They will kill the infected cells and will recruit other immune cells that contribute to the elimination of the infected cells. So antigen-specific T cells are crucial to mount an effective immune response in the body. And for this reason, it's important to detect them in order to have a clear snapshot of the ongoing immune response. So at Immudex, we have developed the Dextramer technology for efficient detection of antigen-specific T cells. So what is a Dextramer? A Dextramer is made of a flexible dextran backbone to which an optimal number of MHC peptide molecules is conjugated, together with a high number of fluorophores. So the high number of MHC peptide complexes that are multimerized onto the backbone increases the sensitivity of detection of antigen-specific T cells, while the high number of fluorophores increases the resolution of detection, and the dextran backbone stabilizes the multimerized MHC peptide complexes. So, but why it is important to multimerize MHC peptide complexes? Because a single interaction between T cell receptors and MHC peptide is very unstable. And therefore, the multimerization of MHC peptide complexes increases the overall avidity of these reagents and allows more stable interaction and therefore better detection of T cells by flow cytometry, compared to other multimer reagents like pentamers or tetramers that carry a lower number of MHC peptide complexes. However, we know that the immune response is highly complex and results from the interplay of several different immune cells. Therefore, a comprehensive analysis of the immune response requires a multi-parametric investigation to be able to identify multiple cell subsets in one sample, to characterize the T cell receptors that are involved in these interactions, and also to define their functional status. And it's for this reason that at Immudex we have developed the decodextramer technology. This technology adds a unique DNA barcode to the dextramer, and the DNA barcode is specific for the MHC peptide complex that is carried on the dextramer and allows the simultaneous analysis of tens to thousands different MHC peptide specificities in one sample by PCR and next generation sequencing. To allow the detection of antigen-specific T cells and also to identify the TCR sequences at the single cell level, we made the decodextramer compatible to the 10x 5' future barcoding technology. This means that the DNA barcode on the decodextramer also contains a capture sequence that is specific for binding to the 10x gel beads. So let's see how the decodextramer reagents fit into the 10x workflow. First, the sample is stained with the pool of decodextramer reagents, each corresponding to a different specificities that we want to look for in our sample. Then, using the PE fluorochrome that is conjugated to the decodextramer, it is possible to sort on the decodextramer positive cells. And this step will increase the frequency of antigen-specific T cells in the sample. The sorted cells will then be loaded into the 10x chromium system to be partitioned into single cell reactions. Then we make 3 DNA libraries, one for antigen specificities, one for paired TCR clone types, and one for the gene expression profiling. Following sequencing of the DNA libraries, we then obtain all this information about T cell antigen specificity, VDJ sequencing, and gene expression profile at the single cell level in our sample. So at Immudex, we have followed this workflow in collaboration with 10x to identify the antigen-specific cells in a healthy donor PBMC sample. In this experiment, we have used a pool of 50 different decodextramer reagents to stain the PBMC sample. And the pool of decodextramer included viral and cancer antigen specificities. So with this screening, we have identified 4 different antigen-specific T cell populations in the sample. 1 specific for influenza, flu, 2 specific for 2 different epitopes of the virus EBV, and 1 specific for the cancer antigen MART1. Moreover, for each identified antigen-specific T cell population, we identified multiple TCR clones. And by looking by the analysis of the T cell receptor clone types that were identified, we observed that viral T cell population showed highly expanded TCR clones, while fewer expanded TCR clones were found for the cancer antigen MART1. So I'm not going to show this data, but it is possible also to further investigate the functional status of the antigen-specific cells identified by looking at the gene expression profiling of these cells. So, but decodextramer technology is not only for T cell research. In fact, DecodClickmer is a reagent that allows to create your own decodextramer by simply attaching antigen molecules to a decodextramer backbone. This flexible technology allows the detection of other cell subsets, like for example, B cells. A DecodClickmer can be loaded with B-cell specific antigens, creating a reagent that is used for the staining of cell samples for the direct detection of B cells. And one of the advantages of these strategies is that together with the detection of antigen-specific B cells, it is also possible to get information on the B cell receptor sequencing and on the genomic profiling at the single cell level. Okay, so in conclusion, decode dextramer technology together with the 10x system allows the simultaneous identification of antigen-specific cells, of their T cell receptor chronotypes, and gene expression profiling at the single cell level. And this offers the full comprehensive overview of the mounted cellular immunity, providing the better picture of the immune response. And thank you all, and I'm happy to answer any questions if you have.
Good morning or afternoon, I guess, depending on where you are. My name is Raquel Deering. I work at Regeneron Pharmaceuticals, which is in Tarrytown, New York. And there I lead two research teams, one that's focused on translational human immuno-oncology work and whose work I'll be representing today. So Regeneron has a sizable portfolio now of immuno-oncology drugs, including checkpoint inhibitors, an increasing number of bispecific antibody therapies in various flavors, many cancer vaccine collaborations with other fantastic companies, as well as CAR T and TCR therapy work internally and in collaboration. And so we're deeply committed to evaluating the biomarker samples from the patients that we treat, obviously to understand how the drugs are working, but also how to find novel targets, shared tumor antigens for vaccine development, to rationally design therapies, et cetera. And so my other research team is actually a preclinical cancer vaccine group. And so it's been quite satisfying to move from the human observations back to preclinical models to be able to develop next generation immunotherapies. So back to the human work, I work with a fantastic cross-functional team in research and development on primarily at this point immuno-oncology trials. And so what we do is implement a lot of protocols to be able to get super high quality longitudinal blood, resected tissues, for example, whole resected tumor material and match normal, and biopsies to run a variety of exploratory assays, including bulk genotyping, bulk gene expression, histopathology, flow, and single cell methods. And those items here listed in or indicated in Rubine, I guess, are those that my lab, my immune monitoring lab particularly oversees. And so we've had to develop a lot of novel therapies to be able to extract as much information from our human patients as possible while reducing the amount of material needed to do that. Even a few years ago, we would bleed people routinely, longitudinally, 80, 120 mils of blood per time point, and now we've gotten that down significantly to often 16 to 20 mils. And so I'll highlight some of the assays that have been developed to make this possible. But generally, there are two core concepts that lead a lot of this work. First, we think that understanding T cell fitness and antigen specificity in the patients will allow us to understand how they're responding to drugs or how they might respond to our drugs. And for the peripheral blood biopsy work, there's kind of a handy trick. So tumor antigen specific T cells do spill out into the blood, particularly while patients are on immunotherapy at certain time points. And so we've been committed to developing next generation technologies to be able to detect and characterize those antigen specific cells in these blood samples. So I'm not a bioinformatician, and the two assays that I'll highlight today were developed in close collaboration with two of my fantastic analyst colleagues, Wendy Zhang and Ankur Danik. So the first is the use of pooled oligotag dextramers to screen reactivities in primary human T cells. The second is a custom internal assay that we call RAPTOR for Rapid T Cell Epitope Ranker. So this does not use dextramers, but can be very handy for finding antigen-specific reactivities. And in both cases, I'm highlighting the technology. I'm not going to show patient samples just due to limitations on what we can show with those materials, but healthy donor blood as proof of concept. So thank you, Stephen and Sarah, for the introduction to the dextramers, the oligotag dextramers. I'm not going to review what they are. That was a perfect lead up, but I will show how we use them. So as I mentioned, we're really interested in identifying antigen-specific T cells in longitudinal blood samples, as well as tumor-infiltrating lymphocytes in the tumor. And then, of course, we often access adjacent normal tissue as well. And so from these starting materials, we have developed an internal process to dissociate samples within the shortest amount of time being removed from the human body as possible. And we like to isolate CD8 positive T cells first. The dextramer reagents are not inexpensive, and the way we stain with them actually uses quite a bit of them. So if we find that if we just know how many CD8s in the population we have to begin with, we can be more parsimonious with the use of the materials. So then we apply these pools of oligotag multimers. We are using between 50 and about 100 oligotag dextramers in our experiments currently. We also apply site-seq antibodies, and so our standard panel of site-seq antibodies at this point contains 250 human antibodies. So we get surface protein information, and of course minimal flow antibodies that will enable sorting, so CD8 dextramer sorting. We then partition into the 10x gems and generate a variety of libraries to be able to capture dextramer, transcript, TCR, and site-seq information. We then deconvolute reactivities using internal pipelines that were developed by Wendy Zhang, as I had introduced in a prior slide, to be able to reveal specific reactivities. So in this example, I know these are a lot of multimers, but trust me, these are a pool of 50 multimers that Immudex and 10x has also used widely that cover a number of human haplotypes. The alleles are given on the left, the minimal CD8 epitopes, and their protein sources. And so this is kind of a control test group of dextramers that we use routinely. And so you won't be surprised to see that many of the reactivities are for common viral antigens, CMV, EBV, influenza, HIV, as well as some common tumor antigens like MART1, WT1. So in this example that I'm going to show, we had PBMCs from a healthy donor, so cryopreserved PBMCs from a healthy donor. His haplotype is here. He was A0201, A29, B35, B57 positive. We haplotype all of our human samples, so we can have more confidence in the reactivities that we identify. And we did kind of every orthogonal functional T cell assay for all of these antigens one could think of, including RAPTOR, which I'll show later. So we knew that he had very high memory reactivities for CMV and EBV and some MART1 as well. So this is what the sort gates looked like prior to our internal staining protocol. And I'm going to show the cell calculations as well because it is relevant. So we started with 60 million cryo PBMCs from this person, isolated 15 million CD8s, performed that series of staining with site-seq and dextramer and flow antibodies, and ultimately sorted about 60,000 dextramer positive CD8s. And so this is what the CD3 by CD8 population looks like. This is a dextramer FMO negative control. So the positive dextramer sort signal is obviously very different and very clear. But even within that sort, you'll notice that we're super conservative with how we sort. So we actually don't even include this lower tail, and that's important later. And so, you know, as it was introduced by Stephen and Sarah, the cells the single cells we ultimately sequence have the dextramer if there was positive reactivity, as well as site-seq antibodies. One technical note. In general, with larger multimer pools, oligotag multimer pools, the total population that gets stained is a bit lower than one would expect if you added up their individual reactivities. So just, you know, just be aware of that. I think we've always seen that in all the samples we've stained. So on the left, this is the positive versus negative signal density by dextramer UMI that we observed in that experiment. So this is the positive signal for any reactivities out of the 50 that were tested. And this is rendered in a way that we have now applied to as a QC to all of our pooled oligodextramer sorts. And I just want to emphasize on this slide why it's so important to develop excellent wet lab methods as well as bioinformatic tools to interpret the data. So we also use this internal method to evaluate the sequencing libraries of 4 donors 1 through 4, that 10x had published in, I think, May of 2019 at this point. And so you can see that for many of the donors, there's actually not excellent discrimination between the positive and the negative. The positive here is in blue, and it really eats into the negative, except for donor 3, which we're going to come back to in the next slide, which actually looks like good discrimination. So this discrimination was achieved by continually optimizing the wet lab's pooled dextramer staining protocols. It is really, really important to carefully develop the wet lab techniques to be able to analyze such complex reagents. So we work freely and have wonderful collaborations with 10x Genomics. So we were able to, for these 2 donors, 3 and 4, receive some of the same exact remaining CD8 material that they used in their wet lab method to generate these original data. And so we applied our oligotag dextramer wet lab techniques and then attempted to sort and sequence using the same exact dextramer pool, sort and sequence at Regeneron. And so for donor 3, the donor that looked like they had a really nice positive population, we were actually unable to achieve a positive population here. But we did get a very nice, good discrimination for donor 4. And this is just the old data for reference. And so when we looked at the donor 3 reactivities from the original 10x analysis, we found that it looked like it was all background. Again, just emphasizing it's really important to learn, just like traditional multimers that are read out by flow, to learn how to use these reagents and reduce background using really excellent optimized wet lab techniques. So coming back to that, other first donor that I was showing, we performed for all 50 multimers, we performed single non-oligo multimer stains as well to compare to what we got from the sequencing. All of those that we were able to get good populations for, we show here in Rubine. So there's very good overlap between I'm actually not able to see my screen so well. Hold on, there we go. So we were able to get very good, I misspoke. So we only did the single multimer staining for 23 of the dextramers that were in the pool. So it was heavily skewed toward the AO2. So we did not have, for example, the B35 single flow readout, but we did find that the majority of the cells deconvoluted by the single cell sequencing were actually specific for a B35 CMV PP65 epitope. We know this patient, this individual is both B35 and AO2 positive, so we expect to see that, as well as what we could confirm with a single flow staining, a high AO2 CMV reactivity. So the other reactivities that are in rubine here, we also had confirmed in this orthogonal flow assay. And so there's very good agreement between the other methods that we've used. We've also done this by ELISpot, et cetera, with the pooled oligo stain. So it's very satisfying to see. We also look at TCR overlap, and just briefly, we find very good exclusive reactivities. So for a given dextramer, deconvoluted dextramer reactivity, say here, this CMV PP65 reactivity, that's the B35, we really don't see overlap with other reactivities. And I'm just showing those that are the most abundant, but this holds true for the whole set of 50 dextramers. So this can be very specific. So moving to another orthogonal assay that we often use in combination with pooled oligo dextramer staining. We can take advantage of some activation-induced markers, often referred to shortly as AIM, that are expressed in T cells shortly after encountering cognate ligand. And so some of these I'm sure you're familiar with include 4-1BB, CD25, CD40 ligand, et And so this is showing in T cells that have a population of about 35 to 40% of their of antigen-specific cells for CMV PP65. We're showing if we just stimulate the cells with peptide only, and then follow the upregulation of these markers over time, some markers seem to reflect the population size better than others. So CD25 and 4-1BB in particular tend to reflect that better. So we wondered if we could use 4-1BB or CD25 as a surrogate for multimers, because we don't always have multimers available. We're interested in CD4s, et cetera. And so here, this is showing the data just in a slightly different way. So this is that same sample of starting T cells that have, as I mentioned before, this population of CMV PP65 positive cells by dextramer. And then we just compare what the re-stimulation with peptide looks at by 4-1BB signal. So it really depends on the reactivity and on the individual, but generally there's relatively good overlap between these two populations. Of course, we would have to look at TCR to be able to confirm that, and so that's what we do here. So coming back to that assay I mentioned earlier, Raptor, we're taking advantage of all oligo-tagged single cell sequencing tools. I'll introduce another one quickly here. And I'm sure some of you are familiar with hashtagging using hashtag oligo reagents. So again, we can really start with anything. Today, we're going to look at PBMCs. And what we do is we distribute our patient samples into individual wells and apply individual distinct stimuli to each of those wells as we would for any other functional assay. We incubate overnight and then apply unique hashtag oligos. So like the oligo-tagged multimers or site-seq reagents, hashtag oligos are reagents, and often they're antibody reagents, although we use lipids as well, that can bind to a marker that's on all cells of the population, say beta-2M or CD2. And they have individual oligonucleotide markers on them so that you can deconvolute where the cell was previously. So here we're using this to be able to pool multiple experimental wells. So we have a hashtag oligo 1 in well 1, hashtag oligo in well 2, hashtag oligo2 in well 2, etc. So all of the cells in these wells will be labeled with their specific hashtag oligo. Why are we doing this? Well, this allows us to then pool all of the samples and hence pool functional signal. So we, after these are uniquely, each of the cells from these wells are uniquely labeled, we pool them, apply our panel of 250 site-seq antibodies, our oligo-tagged dextramers when we want that information as well, flow antibodies, etc. But what we sort is not the dextramer signal in this experiment. What we sort is that AIM, that functionally induced marker. And here we wanted to sort by CD137 or 41BB as it's also called. And so we have in this CD137 positive population, all of the reactive cells in the wells where there was cognate ligand and upregulation of CD137. And so then we can, just like in the pooled oligo experiment, partition and get all of the site-seq, multimer, TCR, and transcript information. And because we can deconvolute the hashtag oligos in the single cell data, we can figure out which cells saw which antigen by hashtag deconvolution. I know this is a bit complicated, but it's super useful technology. And so here's a very basic example of what this deconvolution looks like. So this was a sample that had cells partitioned into 5 different wells, stimulated overnight with individual peptides indicated here that are from common viral reactivities. So EBV, flu, CMV, and then these 5 wells were hashed individually, then pooled and applied to single cell sequencing. The hashing deconvolution looks like this. So these are all individual cells in each of these columns, and yellow is a positive signal for a given hashtag oligo. And just as we saw in this sample by 41BB upregulation by flow, the relative population size for these populations holds. So by far the most frequent reactivity was CMV PP65, and we see that here has the most number of cells. Both EBV BMLF1 and flu M also had sizable populations, and we see that, but we had very little EBV YVL9 reactivity, as we saw by flow as well. And so this is something we now apply to hundreds of reactivities in a given sample, but much more complex than this. But what's really nice is that the use of these hashing reagents allow us to maintain relative population sizes. So not surprisingly, we wanted to compare, and often do side by side, pooled oligotagged dextramer and raptor experiments. So this is a bit smaller than the first experiment, but we applied 23 peptides that correspond to different reactivities across haplotypes, as well as 2 negative controls in both raptor. So this is stimulating cells overnight with each of these peptides and then hashing and pooling for later computational deconvolution, or just directly thawing the material, the cells, isolating CD8s and applying these 23 dextramers directly, and then comparing the TCRs and sequencing information that comes from these 2 orthogonal methods. So this is a representation of the correlation between the TCRs that were found in the dextramer screen and the 41BB raptor screen. And what we found is clone sizes on average for most reactivities, clone sizes in which there were at least 20 cells correlate really, really beautifully with these 2 methods. However, for a lot of reactivities, clone sizes that are around 10 also have tremendously good correlation. We'll see that there are a lot more TCRs that are identified by dextramer than by Raptor. So what does this mean? We actually expect this. Dextramers are a binding assay. They're telling you which TCRs can just bind to cognate epitope, cognate ligand. It doesn't mean that there's going to be a functional reactivity to this binding. It's just telling you what the TCR can recognize. Whereas Raptor is giving you both specificity, but also functional response. And so, so we almost always see that there are more cells that can bind dextramer than those that can functionally respond. And this is just a different way of looking at the data for a few of the other reactivities that were positive in this screen, but overall very good agreement. As I mentioned before, we routinely apply many site-seq antibodies to both the dextramer screens and the Raptor assay, as well as get very high quality transcriptional information at single cell level. And so I don't think we have time to go through this, but this is just an example. And in summary, I will tell you that these reactivities are phenotypically make sense. They're enriched for memory reactivities, but also have revealed a lot of interesting phenotypic states. So in conclusion, I'm blown away by the new single cell technologies that we can apply to real human samples. This has given us tremendous flexibility in what kind of single cell resolution we can get. We can now identify ultra low frequency antigen specific reactivities in all sorts of human tissues at very high resolution. And two of our main assays that we use to do this are the pooled dextramer and Raptor assays. There are advantages and disadvantages to each, and I'd be happy to talk more about these approaches. Feel free to reach out to me. Thank you so much for all of the folks at Regeneron, at Immudex, and 10x Genomics that have been involved in this work.
Thank you so much, Raquel. Fantastic data. Thank you so much for sharing with us.
Absolutely. Are there questions?
Yes, there are a few questions. So I'll start with, do you see this characterization of T cells having utility on clinical trials as a measure of response?
Yeah, so that's actually how we use the assays. I'm just not permitted to show those data, and they will be published, you know, hopefully over the course of the year. But that's absolutely the case. So we want to be able to isolate lower and lower blood volumes. And of course, we do a lot of neoadjuvant trials where we resect the whole tumor as well and correlate tumor antigen specific reactivities with clinical response, which we are doing, and we are seeing interesting correlations, absolutely.
Fantastic, thank you. And what would you say is the average time to set up this kind of workflow?
Yeah, it's a big effort, I'll be honest. So as I've mentioned before, the wet lab methods are as important as the bioinformatic efforts, and they're separate, and they require a lot of QC, and these are, you know, these are expensive readouts. So it does take a little bit of tenacity to get them working, but the amount of information and the quality of information that you get is worth it. And so I would say spend a lot of time optimizing both, get connected with folks who are very experienced in both, and work together back and forth, because what you do in the wet lab, you can then see how it affects in the sequencing data, and it's, you know, an efficient loop. What has been, you know, I keep talking about how expensive the reagents are, but what has been transformational are the availability of hashing reagents, which we now make internally heavily, so we have hundreds of them. So if we go back to the pooled oligodextramer example, that was just one sample. We now redefine what one sample means. So when we do clinical tests, we usually pool up to 20 samples, because we've individually labeled each with a hashtag oligo, and then we apply the pooled dextramer. And that has driven the cost down tremendously. So if you want to use the reagents like this, then in the development, I would suggest also optimizing the hashtag oligo reagent staining and bioinformatic demultiplexing methods. But it's, you know, it took us probably a year of solid work to get very good signal.
Mm-hmm. Thank you so much for sharing that. And I think, Sarah, this is for you. Why do you need your barcoded B-cell specific antigen in multimeric form?
Okay, so you want to multimerize your antigens into a dextran backbone in order to increase the avidity of your reagent. So you create a very stable reagent, a very stable interaction between your antigen and the B-cell receptor on the B-cells, and increase the time of interaction. Therefore, you increase the chance of detecting your B-cells also for very, let's say, low affinity interactions. So this is why you want to multimerize your antigens. And also you want to you have a multimerized fluorochromes on the decode clickmer, so you will also increase the resolution of your detection.
Excellent. And I think another question for you. How many barcode molecules are there on a dextramer?
We cannot disclose this information. I'm sorry, yeah.
Okay. How do you make sure that the peptides bind to the MHC?
Okay, so we have some optimized assays to measure the binding of the peptide to MHC, and we use them as quality controls of our reagents before we actually make the reagent ready for our customers. So, but again, I cannot go into the details of the of our assays. Yeah.
And there is another question, Raquel. I'm not sure whether you can disclose that. How much dextramer do you use at Regeneron?
How much dextramer for like a single stain or just generally?
This is the question exactly. How much? So.
A lot is the answer to both.
Maybe for
a single stain.
For a single stain, yeah. So I think that has required such optimization. I'd have to check to see whether it's something we can share. But what I would say is it's more than what's recommended. And for those of you who are interested in using these reagents and are much more experienced with single dextramer staining workflows, we found that using the pooled dextramer reagents more like what you would do for a single stain in terms of concentrations of cells and reagents, gave us better signal. So I hope that's helpful.
Thank you very much. And another question is the number of antigens on the dextramer backbone always the same, Sarah?
That depends on the reagents. It depends on the reagent, yeah. So from the same reagent, you will expect to have always the same number, yes. But it can vary from one, for example, MHC allele peptide to another one. But yeah, it's very minimal variation. But for example, buying the same reagents over and over, you will expect always the same, yeah.
Excellent. And another question, do you also provide MHC2 carrying antigens? And how do these compare to the MHC1 ones?
Yeah. So we also provide MHC class 2 dextramer, and we have a growing list of MHC class 2 alleles available. And they are also available as decode dextramer. So, and we have also already some data showing the detection of CD4 positive antigen specific T cells, and they are available on our website. And they compare they equally good in detecting the antigen specific T cells, and also the per TCR clonotypes of the populations. Yes. And we have all our available alleles on the Immudex website for any information. But there are also, just wanted to say that if someone is interested in MHC alleles or peptides that are not currently listed in our catalog, we also do custom products. So we can have more than what we already have on our catalog upon request.
Excellent, thank you so much. So we have reached the end of today's webinar. I think we have answered all the questions. So if you have any more questions, please feel free to reach out to us. Thank you so much, Raquel and Sarah and Stephen, for the presentations and all the information that you shared with our audience today.
Thank you for organizing.
Thank you.
Please also join us tomorrow when we have the next webinar of this series with Pavel Zajac on sequencing with Illumina, and Pierre Milpied on deciphering B-cell lymphoma heterogeneity. Thank you so much for joining us today. Have a nice rest of your day.